Hello viewers, this is my part 6 of angiospermic reproduction chapter. So today we will discuss the fertilization. Today we will discuss the fertilization process. So we can write also here double fertilization and triple fusion process. So in fertilization what happens? Before that we can discuss different steps like first pollen pistil interaction, second entry of pollen tube into the ovule. Next, entry of pollen tube into embryo sac, and last fusion of gametes. So first thing is pollen pistil interaction. So here what happens? The pollen falls on the stigma. Pollen falls on the stigma. Here pollen falls on the stigma, and here the stigma recognizes the pollen and the pollen germinate into male gametophyte, where the generative cell divides into two male gamete, and here the tube nucleus is there. So here recognizes the stigma and the pollen germinates. What we here call this is the male gametophyte. So here it will gradually movement occurs because of chemotropic movement, because boron calcium ionocetal is the chemical substance which drag the pollen tube towards the ovule. So in this way you can draw the structure of the ovule, and I have already discussed in the previous videos. Here the antipodals, secondary nuclei, and the egg apparatus is there. So gradually what happens? In this way pollen pistil interaction occurs. Due to chemotropic movement, the movement of pollen tube occurs. The boron and calcium plays vital role in drawing the pollen tube into the style. Next step is entry of pollen tube into the ovule. So this pollen tube may enter in this direction, or may enter through the micropillar end. When it enters through the chalazal, this is called chalazogamy. Chalazogamy. And when it enters through the integuments, this is called mesogamy. And when it enters the micropyle, it is called porogamy. But really what happens in different species, different way of entry of pollen into the ovules takes place. So here maybe chalazogamy, maybe mesogamy, maybe porogamy. Then after entry, whether chalazogamy or mesogamy or porogamy, suppose we can discuss it is entering through the micropyle region. So this is called porogamy. Then it enters into the next step, that is pollen tube entering into the embryo sac. So after entering into the embryo sac, really the synergid plays vital role, having the structure filiform apparatus. This filiform apparatus guides the pollen tube, and pollen tube pour down the two male gametes into the embryo sac. So one male gamete fuses with the egg. One male gamete fuses with the egg plus one male gamete. The process is called syngamy. Syngamy results into zygote. This is 2n, this is n, and this is n. This is 2n, and zygote in future gives rise to the embryo. Then second male gamete fuses with the secondary nuclei. So second male gamete fuses with the secondary nuclei. This is n and this is 2n, results into pen. This is 3n, primary endosperm nucleus. So this pen in future gives rise to endosperm. Which is triploid in nature, and this endosperm is the nutritive tissue for growing embryo. So in this way what happens, so two male gametes separately fuses. One fuses with the secondary nuclei, one fuses with the egg. So it is called double fertilization. And the pen which is produced due to fusion of three nuclei, that is one male gamete plus two polar nuclei, which after fusion name as the secondary nuclei is called triple fusion process. So from this we concluded that the double fertilization and triple fusion, and so finally we can tell syngamy results into zygote, and triple fusion results into pen, or we can tell PEG also, primary endosperm cell, which is triploid in nature. Pen is 3N and zygote is 2N. So this concludes our fertilization process. Then we will discuss how the endosperm development occurs. Development of endosperm. So the development of endosperm may take place by three ways: pre-nuclear, endosperm, cellular endosperm, halovial endosperm. So in pre-nuclear endosperm, what happens? Normally we can see the structure. There is division of nucleus occurs without cytokinesis, or we can tell the karyokinesis is not followed by cytokinesis. This is called pre-nuclear endosperm. And in cellular endosperm, the division of the pen occurs, or the primary endosperm nucleus occurs. First division is like this, and there is cytokinesis. Next division. So in this way, like normal cell, it will divide and produces the different cells or constituent cells of the endosperm. So this is karyokinesis is followed by cytokinesis. So this is the example of kernel of coconut, and this is the coconut water is the free nuclear endosperm. In halovial type, what happens? Normally first division is cellular. Which means the karyokinesis is followed by cytokinesis. The partition wall is formed, and in further divisions, this is free nuclear. First division is cellular, and in further division it is free nuclear. Or we can tell this is the combination of free nuclear as well as cellular types. So in this way, the endosperms are produced, and these endosperms behaves as the nutritive tissue, and it is consumed by the developing embryo. So we can just revise the double fertilization, triple fusion finally by drawing the distinct structure of the embryo sac. This is antipodals, antipodals. This is polar nuclei or secondary nuclei, and here the egg apparatus, ovum, and these two are synergids, and this is the filiform apparatus. So when pollen tube enters, it gives the two male gametes. One fuses with this and another fuses with this. So this is first male gamete and this is the second male gamete, and here this first male gamete and the ovum, this is the process syngamy, what I have discussed also, and this is the triple fusion process results into pen, and here syngamy results into the zygote. So this is all about the double fertilization and triple fusion. In next class we will discuss the development of embryo. So next will be the 7th of my series.